All right, today we're pulling off a kind of scientific heist. But we're not after gold or jewels. Nope, our target is something way more valuable. The secret's hidden deep inside a living cell. You ready? Let's dive in.
So, just think about a cell for a second. It's like this tiny, bustling city, right? It's got power plants, factories, highways, the whole shebang. So how on earth do you study just one building in that city, say the mitochondria, without causing total chaos?
Well, the answer is this really clever technique called cell fractionation. And this isn't about just, smashing the city with a hammer. It's an elegant, super precise way to take it apart piece by piece so we can get a good look at every single component.
And just like any good heist, this operation has a plan. A very specific, three-step plan to break in and sort the goods.
Okay, so here's the game plan. First, you got to get the tissue sample. That's the extraction. Then you crack the cells open, which is called homogenization. And finally, the really cool part, you sort the loot. And that's all done with something called centrifugation.
So that break-in, the homogenization part, it all starts by gently cracking open those cell membranes. And I mean gently. This isn't some chaotic smash and grab. It's all done in this special solution that's designed to protect everything inside.
And why is this solution so special? Well, for one, it's kept super cold. We're talking ice cold. This stops any rogue enzymes, like the ones from lysosomes, from basically just eating up all the valuable parts. And it's also what we call a buffer, which means it keeps the pH perfectly stable. It's like a safe house for all the organelles once they're out in the open.
Okay, now for the main event. The cells are open, all their parts are floating around in that protective solution. Now it's time to sort the loot. And this, my friends, is where the centrifuge comes into play. And honestly, the best way to think of a centrifuge is like a super-powered salad spinner. Seriously, you spin the sample at these insane speeds and physics just takes over. It separates everything inside based on its size and weight. The heaviest, dense stuff gets flung to the bottom first. Simple as that.
But here's the key, the real tactic here. We don't just spin it once and call it a day. We use a method called differential centrifugation. Differential just means we do it in stages. We'll spin it at a low speed to get the big, heavy stuff. Then we take the leftover liquid and spin that again, but faster. And we just keep repeating that process, faster and faster, to separate smaller and smaller parts.
All right, round one. Let's fire up the machine. We're starting with a pretty low speed spin.
We're talking speeds up to 15,000 RPM. Now, to give you some perspective, that's like 20 times faster than your car's engine when it's just idling. So yeah, it's moving. And success! After that first spin, the heaviest and densest parts of the cell, things like the nuclei, get forced right to the bottom. They form this little pellet that we can just scoop out and study. First part of the loot secured.
But we are far from done. We carefully pour off that leftover liquid, the supernatant as it's called, and stick it back in the centrifuge. Time for round two. And this time, we're cracking up the speed.
Okay, now we're pushing it up to 25,000 RPM. You can just imagine the G-force is getting way, way stronger now. And that extra force does the trick. It pulls down the next heaviest components. So now we're collecting things like mitochondria and even some of the cell's little protein factories, the ribosomes. Another layer sorted.
All right, it's time for the final round. We take whatever liquid is left and we are about to hit it with the maximum power of this machine. This is how we get the tiniest, most hidden treasures. And at this point, we're not even just talking about speed and RPM anymore. We're talking about raw force. We measure it in RCF, or relative centrifugal force. And get this, the machine is now generating a force that's 1.5 million times the normal force of gravity. I mean, that is just a staggering amount of power.
So, what's the result of all that incredible power? Well, we can finally pull out the really small stuff. We're talking the tiniest ribosomes, and even individual macromolecules, like strands of DNA or single enzymes. And just like that, we've successfully sorted the entire contents of the cell. The heist is complete.
So, we did it. The heist is a success. We've cracked open the cell, and we've sorted everything out nice and neat. But you might be asking, what's the big payoff here? I mean, why go through all that trouble?
Well, this is where it all comes together. This is the why. Because when you can isolate each part, you can study it all by itself. You can figure out its function, you can analyze ribosomes to understand exactly how proteins get made, you can examine DNA and enzymes up close. But maybe most importantly, you can see what happens when things go wrong. This technique is absolutely crucial for understanding diseases. Researchers can pull out just the mitochondria from cancer cells, for example, to see how they're malfunctioning. This is literally how we develop new targeted drugs, by first understanding the exact, tiny part of the cell that we need to fix.
So in the end, cell fractionation basically gives us the keys to the entire cellular city. We can pick any building we want, go inside, and learn all of its secrets. It puts this incredible power in our hands to explore the very foundations of life. So the big question I'll leave you with is, what should we explore next?